Okay. So, terminology. I already briefly presented this to some of you on Tuesday. Protein-protein interaction. What I mean is that two proteins touch each other. Then I say that the green here, GP120, and the blue, the CD4, are interacting, are in contact because they contact each other, right? In this image, although this is a sort of long-term complex, antibody 2 and CD4, in my view, are not interacting simply because they don't touch. Again, that may be trivial, it may sound trivial to you. In most of the literature that talks about physical interaction, they would say that everything that is at some time point together in space is a physical protein interaction. Okay, this is a unique perspective, and in fact, most publications on protein-protein interaction would actually also talk about association. Example here. A activates B, B activates C, C activates D. Most people would say that all four interact. I would say they are associated. Again, these two things are not physically interacting, these two things are. The typical thing by which you do it experimentally to find out that two proteins interact in a high throughput manner is by yeast to hybrid. By now, there are very, very accurate yeast to hybrid scans. Essentially, you have a reporter that you tag onto your protein X and your bait Y, and if X and Y come close, then there is some protein expressed, a GFP, for instance, simply because of these two, the transcription factor and the activation domain. And you see a signal, and ultimately, this way you scan the entire yeast or the entire human. There are many of these tests, but we still do not have a complete version of all the interacting proteins in human. How far away we are from that after 10 years, over 10 years of yeast to hybrid, at this point it's not even clear. We're not sure whether we have 1/10 of what we are expecting or whether we have 1/2. It's most likely more in the ballpark of 1/10. Okay, now back to the simplest view on protein interaction prediction would be through homology-based inference. Again, homology-based inference is you have a sequence similarity between a query protein for which you do not know the annotation and an experimentally known protein X that is above some threshold. Then you say, I simply take the annotation from X and put it to Q or copy it to Q. Now, the same thing you will do for protein-protein interaction is, however, getting slightly more complicated in the story. So now you no longer have a simple relation, you have pairs. Because now the experimental X is A and B. It's two proteins that bind. Okay? Essentially, you know X in this story here becomes an interaction between A and B. The pair that you want to predict, A prime and B prime, the Q in this one here, is now a pair. And the similarity is also no longer trivial, the threshold T here, because now essentially you have two similarities. You have A and A prime is one sequence similarity, and B and B prime is another one. So you could say I'm only looking at the cases where these two, B and B prime and A prime, are exactly the same. What's the problem with that one? If you restrict homology modeling to situations where this X, A prime, and B prime is identical. So I should have called them X and Y. What's the problem if I do that? Well, you don't have many of these cases. How realistic is that? That you find another protein B prime that is exactly the same sequence identity as the protein A and A prime, and then for both of those you want to predict interaction. So this is unlikely that you find cases like that. So what you could do is you could, of course, what could you do? So ultimately we want a relation larger than some threshold T, but you have two thresholds. How could you handle that? Yeah, I believe you already said.
You can just use a two-dimensional threshold or something.
Yeah, but how? So that actually is so funny. 2 is not much bigger than 1, right? But it makes things getting twice as complicated, and often that is too much. So let's stay with 1. Yes, you could do that. You could essentially think about it as a vector term, but it's amazing how many things get suddenly complicated. Simple way. There are at least 3 simple ways in which you can do that. Yeah?
Can I make a mean?
I'm not saying it's the simplest way. It's one simple way, yes. It sounds very simple, right? Another one is you look at the min. you say that if one of them is very close, that's good, but the sort of, or the maximal difference, so that they can only diverge that much, not more than that, okay? I will not allow that, so maybe this is 90% and this is 80%, but the 80% is giving the call because the most distant one of the two is the one that counts more, you can argue. Or you could argue that the one that is most similar counts, but most likely the most distant is relevant. So if you have a relation between A and A prime that is so distant that I cannot really recognize that they are related, in that extreme case, it doesn't matter how similar B and B prime are, right? The sort of the weakest chain or the weakest link in this chain ultimately is calling the shots. So these are two simple ways of doing it, okay? And this max is what I'm going to show in a minute in the next plot. So the first question is, can we do homology-based inference? And clearly the answer is we can. Otherwise we would never do yeast to hybrid systems, because who cares about yeast? Ultimately, we do yeast to hybrid systems because we hope that studying proteins in yeast, we can learn something about proteins in human, because we assume that this works, right? Okay, let's just look how well it works, and the scale here is the Hspeed threshold. Again, the curve blue here is distinguishing things that are above the curve have similar 3D structures, things below the curve we don't know. This is number of residues aligned, percentage sequence identity, plus an h-val plus 5, plus 10, plus 15, plus 20, minus 5, minus blah, blah. Here we anyway have only plus distances plotted here. And the first result that you see is that for a high level of sequence similarity, again that's the max, so the most distant one of the two, we have something, and for the lowest ones that I plot here, we have something else, and there's some difference, but I'm sorry, but given the error estimates, this is not even significant. So it's fairly flat. The next feature that you observe here, well, high error rates, but the next feature that you observe is that these numbers here on the y-axis are very small. This actually is precision, or what we typically call precision. Why is it that low? Well, ultimately what we do here, and this is work that is a while ago, 12 years ago, what we would do today in the same way is we would simply take all the protein interactions that we experimentally know, would say if something is not known and I predict it is wrong. Now, because it hasn't been observed doesn't mean it's not right, but you cannot do it differently. If you want to compile precision, if you want to compile anything on this axis, you have to have some that are wrong. Right? And you have to, the only thing you can do at the moment today is call everything you don't know yet not existing. Okay? So meaning wrong, and that sort of explains why this value is so small. But it is seemingly true that there is some drop. It is unclear whether this drop is sort of like the kind of T's that we saw for structure in green, for localization in blue here of an enzyme, depending on whether I know 4 or 1 enzyme number. Is that a similar drop? We can't tell because of the error function. But there is another issue here. And that issue is the issue about sort of homology. Typically homology comes from, as I mentioned before, organs, or the similarity on the level of organisms and organs. So, sorry, the homology is between the hand and the fin of the whale. Although that when you see the whale, it's not recognizable that this is actually hand. But when you sort of look at the detail, you see the homology immediately. And you see the homology even for birds that you may typically not sort of feel are just like your hand and your arm. But on the level of the genes, ultimately the way we see that is the genes, or the proteins, here in this particular case species A has for simplicity 3 genes or 3 proteins. Then there is a speciation event. They both essentially have the same set of proteins. Upon the next speciation event here, there's a duplication of a domain, and then you have this in the 3rd generation, something like this, and then the problem becomes orthologs, paralogs. So ultimately there is a domain here that is duplicated and changes, because already you have sort of this domain doing the function, so you don't need to keep the one that looks exactly identical, so you have the freedom to change. That is the idea. And all of that, this is all simple, But it gets, of course, complicated the moment you have jumping genes. You do know that we have jumping genes, right? Everybody knows that. Do you also remember where it was found first? How long ago since when do people know that there are jumping genes? Any idea? Do you have any idea when this idea of gene comes up? Genetic material.
With Mendel?
Not quite. So, because at this point we are not, yes, so there are the rules of the way we can essentially do it in practice. But we don't at this point have the idea that there is a genome that is inside of a protein, inside of a cell, in the nucleus, and that is the DNA, and that the DNA leads to proteins. This, at that point, is not clear. There is some idea that there is some genetic material. I mean, it's not called genetic material at that point, but that means that at the Mendel time, you still don't have this idea that there is an element called gene. That is actually way later. And so this is essentially, we are talking about less than 100 years. So ultimately the genetic code is the 60s. Fitch. Well, that's the 3D structure of the genetic code, yes. True, correct. Not the 3D structure, yes, that is the, but the idea that you have, okay, so genetic code has different aspects of it. Anyways, so the triplet and blah, blah, blah. Jumping gene is actually something that happened before that. This is 40s, and it was first discovered in corn. And essentially, so one thing that you may all have seen is corn that has many colors. Multicolored corn. So when you go to the supermarket, typically you see this yellow corn that you can eat. But then you sometimes see, in particular in Mexico, on the Day of the Dead or Halloween, you see these very colorful corn. And they, in fact, are the result of jumping genes. And you can create them, and that is in fact the first time they were observed, to see that one color was sort of imported into the other plant by a jumping gene. Here we have an organism, an algae that incorporates an entire chloroplast. So the sea slug incorporates an entire chloroplast and makes it work. This is an example where you do not only have one jumping gene, you have much more exchange. And this is very common, and that makes it more complicated to really see in trees what is an ortholog and what is a paralog. What is what comes in from somewhere else and what is a duplication event. It's very difficult to see. But ultimately the question arises, for my homology-based inference, the story between orthologs and paralogs. Or let me explain it differently. So assume I had a second pair. So I have these two, I know that they interact, A and B. I have one pair, A prime and B prime, that I want to know whether they interact, and one other pair, A2 prime and B2 prime. Now A prime and B prime are in the same organism as A and B. A2 prime and B2 prime in a different organism. And I choose my case such that the sequence similarity, or the maximal distance of any of those, is the same for A and A prime, and A and A2 prime. So they're equally distant. I'm going to pick cases where they're equally distant, and I'm going to ask you, for which of these two cases homology-based inference is going to work better? If I have the situation within the same organism, or if I do between two different organisms.
I would argue between two organisms, because
yes.
If it's in the same organism, they will probably evolve and have different functions.
Absolutely. This is absolutely what people expect. This is absolutely the biological intuition. That in fact this is the model that we continue to absolutely believe in, that new function is evolving through duplication events where the existing gene is there and the new one therefore can do something new because the other one is there already. Right? So this is a duplication of A inside of the same gene, and because it's a duplication it means that A is there. There is no reason to maintain this interaction because it is already maintained with the original ones it was duplicated from. That's the entire idea of evolving new features through duplication. And that, in fact, was, again, so here we have, that was warm against versus non-warm. So that's the case of orthologs, if you want, between organisms. And that is the case within the organism. So that's paralog. And again, this is one of those papers that was very difficult to publish because it's exactly the opposite what every reviewer and everybody in the world believed. And continues to believe. So it's published in 12 years. People have repeated the same result in many publications. And they have now shown it also for other features. It's a contradiction to the biological understanding. And we don't know what it means. We have absolutely no explanation for it. It's an observation. By now, I believe everybody accepts this observation, but we still actually do not know what it means. Yes?
But can't you explain that it's close? Because if it's within the same species, then you should have more similarity because the whole surrounding, the whole system is still similar. And if you have a different
but the biological argument really is you evolve new function. So, Michaela, so the first thing that I believe I'm always, what makes me very nervous is you have to always, when you do an experiment in a computer, you have to always sit down for a moment, reflect, and predict what will come out. And then you have to reflect on whether you saw that or not. Typically, the problem, I believe, is when you expect A to come out and you observe non-A. Be very careful, in my view, to explain why you always expected non-A. I've seen it again and again, that people, when I ask them, they say A, and then I show them, yes, the result is non-A, and then they answer, yes, well, I always expected non-A. We have to be very, very careful with that. So this is a very, I don't want to set you up, but it's a very important thing that I feel we as a community should learn. And I'm saying that because I've played this kind of game, or this kind of question-answer game with many of very well-esteemed colleagues. And we all tend to, in the aftermath of the effect, suddenly know why we actually always expected, yeah, true, but it's the other way around, right?
but actually before we...
But let's go back to the reality. So when you go into rechts der Isar, or when you go to any hospital, right, you take mouse systems. Why? Because mouse has a different environment than human? No, actually not, right? You essentially assume that the cellular environment in mouse is very similar. So this was warm versus not warm here. That is fly. So fly. Okay, fly and warm are not good examples. But I would still argue that when you look at the local environment of a cell, maybe it's not that different. And if it would be that different, we have to really begin to wonder whether our mouse for human experiments are relevant. Well, we believe somehow that they are relevant, right? In that sense, I believe that's the limit or the constraint to your point that the environment is different. But ultimately, Michaela, you're right. The observation is that way. So there must be something in that direction. Maybe, so one way of saying is maybe there is an increase. And again, it's not only for protein interactions, this is also for Go function, this is also for enzymatic activity. So maybe there is an advantage if you somehow keep this old function around. Maybe it increases the probability if something does go wrong with A, and you have the backup interaction, maybe that does help you. Maybe there is something that is in protein-protein interactions that you defend yourself against sort of importing other interactions. Maybe you're right. Anyway, that's the observation. As I said, we don't quite know why. Does it mean that paralogs are conserved in interactions, orthologs are not? Certainly not totally, but what is clear is that this idea of model organisms is getting complicated. Now that brings us to a different question here. Let's assume that I measure the same interaction. Now, we have not the situation of homology, we have the situation of A and B binding. And I simply do observe the same protein-protein interaction, A and B, twice. Okay? So now it's no longer the similarity here, it really is 2 times measuring the same thing. 2 different X-ray experiments. So what do we expect in terms of the interaction? Typically what we expect, of course, is that we, within the experimental error, observe the same interaction. And that turns out that actually it's not that easy to measure what's the same interaction. How would you measure that it's the same interaction? So I'm asking, if I do the experiment twice, do I have this? It's not really the interaction of the pairing. I'm really looking at the detail of the interaction. But how would you measure whether the details are the same?
Binding energy.
One way. Yes, you would, for instance, say, I have a 3D interaction. And in this 3D interaction, I know that, say, these 3 residues bind. I have a type of binding, and I'm going to define a model by which I call this, by which I define an energy of binding, or the free energy of the interaction itself, right? We can do that. That's model number 1. What else could we do? It's not that simple, but in fact, we did that. What else can we do? So I'm saying it's not that simple is because ultimately there are different models that you can compute. Computing binding interactions always is the question, do you compute the binding interaction for the 2 proteins as they are? Or do you compute the binding interaction in water? Do you compute the binding interaction from the proteins as they are not binding? And that, in fact, you can't do, as I briefly explained the other day, or mentioned the other day. So the binding is sort of an approximation. But what could you do that is sort of similar as a proxy measure? That is not really an approximation, but it's also less meaningful. Any idea? What could you look at? So we want to know whether 2 proteins always bind, say this way, or sometimes this way or that way. What would you measure? Yeah?
Could you compare the sequence similarity and the motifs or something?
So, not sure what you mean by the motif. So again, we have 2 identical proteins. So we have the protein A binds A, binds B, and protein A binds B. So that's number 1, and that's number 2. It's 2 different measurements, but it's the same pair. So what exactly do you? So A and A are 100%, and B are 100%. What exactly do you want to measure? What's the binding motif?
I don't know. I mean, we said binding energy. I don't know if it was just the qualitative, like which bases, which amino acids, for example, bind with each other.
Okay.
And we also want to see if they have the same energy, the same.
So we could simply look, are the same amino acid types used in the interaction? So in the interaction between these 2 proteins, that is A and that is B, I have some region where they bind. That region I use to define an energy, and that region I can simply label and say, in this particular region, we have 1, 2, 3, 4, 5 here, and 1, 2, 3, 4, 5, 6 here. So those would essentially be the base, and I would ask, are these the same residues? The same residues, so there are 2 different ways in which I can do that. I can ask, are these residues number 41? Right, 40, 41, 47, 48 or something. And I can say, are these alanines? Those are 2 different 2 different conservation scores, right? I could say maybe it sort of moves around a little bit, but we still have a salt bridge. Maybe it's a different salt bridge, or we have a cysteine interaction or something like that, right? So we could have amino acid type, we could have amino acid number, we could have bind energy. What else?
Can we measure the solvent accessibility?
So we can essentially measure the size of the part of the interaction that is essentially the water that is pushed out, or the size of the interaction. So the displaced accessibility is one way of measuring that. You can essentially also measure the accessibility that is in the interface, but ultimately what you would look at is the change in solvent accessibility upon binding. This is one way of measuring it. So now we have 4. Somebody, another one?
secondary structure information.
Yes, we could do that, but that's indirect, right? All of these are direct measures. All of these are... The fact is that we haven't ever looked at that. That's an interesting question. But all of these are... If I see a difference in the secondary structure, that could happen, that could be... Well, yes. We haven't done it. Actually, yes. We could have done that, yes. Could have done that. Something else? I cannot argue against it. It's an interesting idea.
Isn't it like very tricky to actually get the secondary structure to?
No. We have three. No, but we have 3D structures here. I'm going to look at samples where, in fact, these are 2 different X-ray structures. In fact, they are all X-ray structures. We wanted to avoid the problem of NMR models and things like that. What else could we do?
I don't know if it's already said with secondary structure, but maybe just the overall confirmation of the complex, not the complex, but if it's a physical interaction, the overall confirmation of the protein-protein interaction. Because then very often if 2 proteins bind together, they have some kind of conformational change and you have different information.
Yes, so in some sense, so the direct way is to change the change in solvent accessibility, right, in some sense. An indirect one would be actually the change in secondary structure. More indirect one, what else could you measure? The RMSD. Yes, that actually is okay, so there are 2, now you guys bring up new ones actually. Essentially an RMSD. Yes, and they both would make sense, and both we didn't do. So another thing, so let's just cut to the chase here. What we also measured essentially is the binding pattern. Is it the same alanine that binds, the same amino acid, not alanine? Do I maintain the contacts? Right? So now we have an issue. I believe we had these 5, maybe we had even more. But we had something such as 5. Now we have 2 possible ways in which we can proceed. We can say that they differ if they differ by all of these criteria. Or we can say they differ if they differ at least in one of these criteria. Right? Now the reality is we found that these criteria, although somehow we assumed they all measure sort of the same thing, they're not correlated. And we found that in fact, do they differ or not depends a little bit what you're looking at. I'm explaining it. One way of saying that is that if any of these measures differ, then we see that essentially in all of them. In every single one of these pairs, AB1 and AB2, we see some difference in some of these criteria. If we want that they all differ in all, then for 10% we roughly see that 10% of the cases that are measured twice disagree by all of these criteria. Okay, so, and here are some examples. So, where in fact these are alternative ways of binding to the green. And you see they're clearly different, right? So this propeller and this propeller is essentially, you twist you twist it around a little bit, but it's very different. These are alternative conformations. The blue one is bound here, here, or here, but they are very, very different. It's clearly, and they are, again, these are experimental observations. Here, again, in these 2 different sides of the molecule. So we do really see that cases are not just a little bit of different, they're totally different in some cases. And those are the 10% of the cases. The 10% of the cases really differ a lot. What does it mean? Welcome to the world of biology. We actually really don't know. This is a surprise. We assume that ultimately these are exceptional cases, but this is also something that is seen more and more often. Dynamics is more relevant than we believe maybe is what is behind it. Maybe it's a probabilistic thing, so that most of the time it's that confirmation, sometimes you observe that one, and as long as most of the time is that, it makes a difference. Maybe that's what the story is. We don't quite know. There was recently a publication where they showed that for an HIV, I can't remember which protein from HIV, the fact that it did bind in many places was essential for the attack of the virus, for a peptide, HIV peptide. Okay, let's go to the simplest way of predicting protein-protein interactions. And the simplest way is this one. I take one protein, I take another protein, I take a machine learning device, and I simply learn, in this particular case, 1, 2, 3 consecutive residues in one protein and 3 consecutive residues in the other protein, and I ask myself, do they bind? And I do a machine learning on that. And in fact, this is the first method that I tried about 20 years ago, and it completely failed. It completely failed, I believe, because ultimately we have this ocean of false positives. Why do we have so many false positives? What is in this model? I have to make the next point. Of course, I take these two windows and I slide them through, right? If I have my two proteins, so in one protein I sort of slide it through this window here of 3 through the entire protein, and I do the same in the other one. So I have if protein number if the length is LA and LB, then essentially the number of windows I see will be LA times LB, right? The square of the two. Why does that create a lot of false positives?
Well, first of all, because we don't take into account secondary structure.
We don't take anything into account, no.
As in like, maybe the two windows are not even, like, one window might be buried in the protein.
That's the crucial thing.
Might be.
So one of the problems, of course, is that this part of the protein may bind. If I put the sequence, stretch the sequence out, they may bind. But in vivo or in vitro in this particular case, since they are not on the surface, they will never see each other. In this model, I cannot sort of account for that, and I will clearly add a lot of false positives. What else is it that I'm doing wrong? What would be other reasons why they may never meet?
The window size might be too small.
No, but this is not an issue for false positives. The number of false positives is the same with a window of 5 or with a window of 55. Yeah?
The data set is unbalanced.
That's true, but it has nothing to do with the false positives. I mean, okay. It does. But what I mean here, so the issue of buried versus, so the buried regions that I put into my window here, and that will never bind, depending on, it's completely irrelevant what the buried, what the imbalanced data set is. That is going to kill me. If I try to make things meet that never meet, no matter what the ratio is, right, this is going to create false positives. And this is the kind of thing that I'm asking. So what other reasons are there to create a lot of extra false positives independent of the ratio? Could you think about something else? So buried versus exposed is one way. Something that we discussed about in this, yeah?
Well,
I should have seen that.
there's residues that might be close together, might not be close in the structure.
Yes, so that is an important point, and we will briefly get back to that, not today, but on Tuesday. So most likely when I do any kind of machine learning, it sort of will put up, and Benjamin is right, ultimately we need a bigger window. Ultimately we will pick up some consecutive signal. If the protein interaction that I'm looking at is finger-like, so the protein chain runs here, 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 and these things bind, and I don't have a consecutive signal, then this will be complicated to pick up. That clearly is true. But that is a kind of signal that will be a killer if that would be dominated. So if all the protein interactions would be like this, it would be very difficult to predict them from sequence alone. This could be true. So it could be that this method failed because of that. Right. So we have to keep in mind that we have to look at the question, how sequence consecutive are these interactions? I'll show you something.
Anything to do with false positives, then, so it would have something to do with false negatives with it. Because, like, you would have the special case. That
We essentially would see nothing, yes.
Yeah.
That's correct. We'd miss them all. That is correct.
I also think, like, for false positives, maybe, like, just because two sequences can bind doesn't necessarily mean that they're always bound. Even
But why?
close.
Why not? What will prevent them? Well, if you put them in a tube and they bind,
Yeah, maybe there is some more complex structure,
such as.
like an inhibitor or something.
So you need another protein in the vicinity, or you need an enzyme that makes the reaction happen. That could be the case. So this is essentially, now we're getting into the issue, maybe it doesn't happen because there's a temporal issue or a local issue. So maybe they're not coming together in the right environment. Now that is also the most extreme one of the right environment is one protein is nuclear, the other one is secreted. In the tube they may bind, in reality they will not, yes?
But this not the same sublocation problem you also have in most of the experimental procedures, don't you?
That you will also have in most of the experimental
So
procedures.
why for you?
No, because ultimately, not completely, because essentially yeast to hybrid is a system on a living cell, so you will not have it in the same to the same degree. And a lot of the more recent these kind of sequencing or these kind of modern ways to probe protein-protein interactions, they are essentially in living cells. Okay, then you grind up these cells.
Yeah, but in yeast to hybrid you use fusion proteins you specifically created.
But in a cell.
Yeah, but they are brought into the same location.
Not quite, but there are issues. But ultimately what you're putting your finger on in this particular case actually is more the problems with yeast to hybrid. So more advanced yeast to hybrid systems do get around that.
Yeah, sure, but you wouldn't see that in your data. Okay, yeah. If you use the old systems of yeast to hybrid, you wouldn't see that in your data probably.
Yeah, you would see some of it at least. Some of it at least. But then, so the other issue, if they cannot bind because the local environment, so meaning that they don't come together, they're both nuclear proteins, but the proteins needed in order to, there must be a transcription factor next to it or something, and that doesn't exist. Or that it's the wrong moment, the other one doesn't exist, is not there yet. All of these things. Okay, but let's look at the first issue. The first issue that Benjamin brought up is solvent accessibility. So let's just illustrate how we can reduce false positives by solvent accessibility by a factor of 4. Essentially assume that there is a core, a surface, and roughly it's half-half. So half of the residues are on the surface. I'm going to freeze you for another few minutes now. The half of it is on the surface, half is at the core. That then means that in fact, what you instead of seeing the entire protein of both ends here, you have only 1/2 on both ends, and that ultimately means that you have only 1/4 of what you could do, which means you throw out three quarters. That means essentially you throw out by the false positive by a factor of 4. It's only 1/4 that is left of the false positives. Okay, in throwing this out, you make mistakes. In predicting that something's on the surface, you lose some. But most of the ones that you lose or that you throw away, most likely are the true cases. And then you could do sort of the same thing by predicting residues in external interfaces. And this is what we are going to focus on Tuesday. Yes?
So this, like how many true positives do you actually lose by that?
Fewer than false positives. That much I can assure you.
Okay.
You will lose some, yes. That's true, but I believe, I would argue this is worth.
So obviously not from the core ones, but if I look at the picture from before, where the protein binds to like some kind of membrane or whatnot, so you would also lose like a lot of those. Like for instance, in A, you would lose like half of the true positives.
No, wait. So the idea essentially is you're going to throw the core out.
Oh,
So you're going to only throw.
That's amino
Yes.
acids. Oh, okay.
You're only going to throw this. So this is a protein. This is amino acids, right? I'm sorry for the confusion. So I should change that slide and only keep that one.
As long as you keep more true positives, a lot more true positives than you throw out, should be okay. I don't know how to ensure that though.
You assume that I bring in more than I throw?
No,
No, I cannot bring in more.
You keep more true positives than you throw out.
No, but wait a minute. So one thing is clear. We do not know how much of the surface is the one that is involved in the binding. What we do know is that the cores are not binding to each other. Okay? Protein-protein binding is a surface thing. So the half that we throw away, if I did not make a mistake in it, almost always, so yes, proteins do move upon binding and they somehow open, but rarely. So if I really think about a simple double model where we have exposed half is in the inside, in that model this is pretty much in the inside, and not many protein interactions will reach that much. Okay, some will. Okay, you will throw away some. But most interactions will happen on the surface. Again, there's still a lot of the other protein might bind on this side only. So all of these here are nonsense, right? That's true. But at least this is 50% less bad than before. Which again, since it's a square effect here, you throw away by a factor of 4. Now if I could actually really chime into the binding sites, that would help me much more, and I will get back to that on Tuesday. Let's assume that I actually have done that, then I can look at localization. That helps me at least somehow. We talked about that. There's this method that helps. Then sort of we can predict proteins that are sort of binding to other small molecule substrate. That's an alternative, a competitive binding, so to speak. And we had something like that a while ago. Maria is redoing that. We have to be better at predicting domains because proteins bind as domains, improve alignments, and then we sort of can put it all together. And the reality is, I've been sort of giving this talk on protein interactions for many years. It took about 10 years to do all of this, with way more than 10 people. And I showed this slide, and I said, then now all we need to do is put it all together. And I put a face in here. And the face changed from every 2 years. There was a new face. Because it turned out to be the last step to put it all together was much more complicated. And this is again, we're going to talk about it at the beginning of next year, why this is so complicated to do the machine learning really on the protein-protein interaction pairing. Tobias is the one who put it together, but it's highly non-trivial. Okay, putting the data together, most of that we talked about. Physical protein interaction is what we are talking about. We're looking at PDB. We do a sequence unique extraction. We say contact is something that is 6.5 angstrom for the closest atom. We map to a Swiss plot to find out what is a transient protein-protein interaction. And here we get to the point I forgot your name.
My name?
Vanessa, no. No, Vanessa's name I forgot. Oh yeah, that could be true. Not that bad. So what I'm showing here is I'm looking at windows of 9 consecutive residues. And I'm asking, in these 9 consecutive residues, how many of those are involved in a protein-protein interaction? So if it all would be sort of the finger interaction like this one here, then in the most extreme case, I would mostly see 1 in every window of 9, or whatever you look at, right? So I have 2 different thresholds. In blue I call something a contact if the closest atom is within 6 angstrom, yellow within 4 angstrom. You do see that these 2 curves are sort of slightly moved. The peak for the more rigorous cutoff is a little bit lower, so fewer residues in the window of 9, sort of 4 to 5, while the other one is sort of 5 to 6. But ultimately you see that for most of these, in a window of 9, most of the time you do not have 1, 2, 3 for both of those, right? So most protein interaction sites, therefore, and this is not based on interaction interfaces, this is really sites. So this is essentially computed for every single residue where I have binding. I do every single residue where I have binding to another protein. I do a window around that residue of 9. Okay? And then I sort of sum it all up over all of these windows. Yes?
So you have nine residues, and for each residue you look at the 9
So
because
I take 9 consecutive. So I find one residue in a protein that interacts with another protein, and interact is defined as blue less than 6 angstrom, yellow less than 4 angstrom. Now I call that interaction, and I do this for every residue in the protein. And I move myself to the first residue. Okay, and now I'm looking at plus minus 4 on either side. So this one plus the 4 left and right of it. And I ask, in these 9, where this one is at the center, how many of those are also involved, also labeled as interacting? And the answer to that is the number here. Okay, so you see that a substantial set of the protein interactions are things that are not these fingers. So the true negative story that you talked about before, the idea that you had, Vanessa. So most of them are somehow more, the way you can picture them is more that you have some well, oh, I had a picture of that above there. So you have some protein here, and you have some region where you have more than one local. And then maybe this folds back here and goes like this or something like that. And this one here is too far away for the window of 9. But it's not the kind of thing where you have this one here in a protein where it's really far away. These 3 here, the 3 dots I make, are very far away in sequence. Yes, you have some of these situations, but most of the time you have things where you have long stretches that are somewhat parallel. Right? Is that clear?
So this is only regarding the residues that actually are within the that undergo interactions.
Yes. It's the sequence. So if you're interacting with another protein, then you're very likely to be in an environment where the residues next and right of you, not in 3D structure left and right of you, but in 1D, just on a Merle sequence level, are also interacting.
So the finger leg would be a very, very...
the finger would be essentially having a peak here. If it would be largely finger, then you would in this...
Why wouldn't the finger be in the middle, because the finger...
No, that's just the number. So in the finger case, you have one interaction in a window of 9. So in the extreme finger here, 1, 2, 3, 4, 5, 6, 7, 8, 9 or something like that, only one interacts. So this figure here would peak up here.